Thank you very much indeed for that very kind introduction and for the invitation to speak here this evening. I'd also like to thank all of you for giving up part of your Friday evening to be here this evening to listen to this lecture. It's a real honor and privilege for me to be here, stood in front of you, amongst so many physiologists and people who just love physiology. I have been fascinated by physiology my whole life, really, human physiology in particular. Biology was by far my favorite subject at school. Physiology was pretty much the only part of medical school I really paid attention to. I did a higher degree, a PhD in applied physiology, and I'm now privileged enough to sort of see human physiology in action every single day at work with the patients that I care for on the intensive care unit. So what I'd like to do this evening is take you on a journey, really. And I want to take you on a journey from sea level to the summit of the highest mountain on Earth, Mount Everest, and back to sea level again, to the intensive care units where I work here in London. And on that journey, what I'd like to do is share with you some of our research, some of the questions we have about human physiology and about patients, and some of the findings we've made along the way. And hopefully I'll be able to demonstrate to you during this journey the real value that there is in using the environment around us to learn more about human physiology and hopefully to help the patients who are severely unwell with life-threatening conditions in intensive care units around the world. So I'd like to start my journey here, and this is my real world, really. This is what we see every day on an intensive care unit. And an intensive care unit has many similarities to a sort of finely controlled experiment when you think about it. We're continuously monitoring patients like this, adjusting their treatments, titrating drug doses, and optimizing their physiology in order to maximize their chances of survival in the intensive care unit. For those of you not so familiar with intensive care, it looks a little bit like this. We look after the most unwell patients in the hospital. Most of the patients that we look after tend to be suffering from what we call organ failure. So one or more of their organs has failed. Heart failure, lung failure, liver failure, kidney failure, sometimes all of those organs at the same time. There's no real defining illness that brings together the patients on an intensive care unit. They have all sorts of different diagnoses that have brought them to the unit. But one thing that does tend to unite them is hypoxia, which is a lack of oxygen, and particularly a lack of oxygen at a cellular level, the oxygen that's required to generate energy that's essential for life. So one of the real issues that we have on intensive care, as I've alluded to, is there's no commonality in the diseases that we treat there. This enormous heterogeneity of illness and of the treatments that we use to treat the illnesses mean that it's very, very difficult to construct meaningful clinical trials to test the outcomes of drugs and treatments in our patients. I don't think I really need to explain to this audience that to construct a robust experiment, what you really need to do is alter one single factor in a complex system and then measure an outcome so that you can see the difference that altering that one factor has made. Now, this is virtually impossible in the intensive care unit, and the reason for that is that patients do not come like this. Each time I do a ward round on the intensive care unit, this is what I tend to see. Every single patient with a completely different disease, but in the end, all suffering from organ failure and all striving to get more oxygen around their body. And what this means is that if you do want to construct a trial, there's a very low signal-to-noise ratio. So you would have to have an enormous trial to prove something to be true, even if you had a very strong suspicion that the treatment or drug that you were looking at would be successful. And this was highlighted quite recently in this very nice review of what we call sepsis. And sepsis is overwhelming infection. And these authors went through the sort of modern treatments for sepsis in intensive care patients, and they highlighted a number of recent drug trials where drugs had been used in order to reduce mortality and improve survival in patients suffering from sepsis on the intensive care unit. And they looked at 11 trials here, shown in this pie graph. Now, you might not think this is too bad. Eight of the drugs that they looked at made no difference at all. One of them quite disturbingly caused harm to patients, but 2 of them worked. So not too bad an outcome. But when you look at how those drugs fared in subsequent trials, neither of those drugs turned out to have worked in the end, leaving no drugs of any benefit. And actually, over the last 30 years, there have been over 100 drug trials in critical care looking at treatments for sepsis, and not one of them has shown any benefit at all other than the use of antibiotics. And to try and sort of show you the magnitude of the problem that we're dealing with in intensive care, these are data that are collected every year, audit data from every single intensive care unit in the country. And this is just the simple mortality of patients who come through intensive care, showing the in-hospital mortality. So if you were admitted to an intensive care unit in this country with pneumonia, so a chest infection, 1/3 of patients will die before leaving the hospital. If you have severe kidney injury, 40% of those patients will die before leaving the hospital, 40% for liver failure, and just slightly under 1/3 of patients for sepsis, the condition we mentioned before. So this is an enormous number of patients dying in hospital of critical illness, and these mortality rates have not really changed over the last 2 decades because we've failed to be able to find effective treatments for the patients that we look after. So I want to zoom out a little bit now from the intensive care unit and think about human life on Earth. Humans are largely confined to a very narrow set of conditions on the Earth, primarily defined by the conditions within which we evolved as a species. So we mainly live on the land, at sea level, and in a tropical environment. And actually, this consists of less than 15% of the surface of the Earth. So we can only really live comfortably on a very small portion of the surface of our own planet. And there are a whole host of other physiological stresses that we encounter in our daily lives. Most of the time, we don't even notice it. Things like the Earth's gravity, its magnetic field, and the inevitable day-night cycle that occurs every 24 hours, causing circadian rhythms within us. But if we drift out of one of these environmental comfort zones, we undergo physiological stresses and pressures on the body which cause changes. And if these physiological stresses are sustained, the human body is able to adapt to these changes. And these adaptations we call acclimatization. And examples of environments which we can acclimatize to include heat, cold, the microgravity of space, and the topic of today's talk, the low pressure that's encountered as you ascend to high altitude. So I'm going to focus on high altitude and the physiology of high altitude for a little bit, just to bring us all up to speed about what happens when one ascends to high altitude. Mountains have drawn explorers towards them for centuries, really, now, and none more so than this mountain, which is Mount Everest, known locally as Sagarmatha or Chomolungma. It lies on the border between Nepal and Tibet, and the summit of Mount Everest, at 8,848 meters, is the highest point on the surface of the Earth. And it's an interesting coincidence, perhaps, that that point, the highest point on the surface of the Earth, also appears to be the limit of human adaptation to hypoxia at high altitude. And we'll show you some data later to confirm that. Now, the main stressor that occurs when we ascend to high altitude is a reduction in the barometric pressure, so a fall in pressure as we ascend to high altitude. And this fall is exponential in nature, and you can see on this red graph on the left-hand side a sort of gentle decline in the pressure to start with, and then a more sudden decline later on, with Mount Everest at the bottom there to give you some sort of scale. And this occurs because air has mass and it's also compressible. The bit that people sometimes find it hard to get their head around is that the concentration of oxygen remains the same throughout the atmosphere. So up to about a level of 100 kilometers above sea level, it's about 21% oxygen all the way up. And that's just due to the random mixing of gases within the air. But when you combine these two phenomena together, the fall in pressure and a stable concentration, what you get is a reduction in what we call the partial pressure of oxygen. And partial pressure, really you can just convert in your head to meaning number of molecules. And you can see a sort of representation of it on the left there. As you ascend higher and higher up a mountain, there are less and less molecules of oxygen in the air to breathe. And just to quantify that a little bit for you, as you ascend Mount Everest, when you get to the base camp of Mount Everest, the barometric pressure is about half of what it is here at sea level, which means the partial pressure is about half. So basically you're breathing half the amount of oxygen, even at base camp, than you are at sea level. And by the time you reach the summit of Mount Everest, if you're breathing the air at the summit, it's about a third the partial pressure of oxygen when you get there. The physiological consequences of this are phenomenal, and it causes what we call hypoxemia. And hypoxemia simply means lack of oxygen in the blood. And I took this photo earlier on in the year at the ALMA telescope array in the Atacama Desert because it very neatly displays some quite smart physiology. Atacama, this array is at about 5,000 meters, and what you can see on the left is two pulse oximeters on two different individuals. Both of them ascended at the same rate to reach the same altitude, and the one on the top you can see is reading 66% oxygen, and the one at the bottom reading 86% oxygen. One of those people has summited Everest, and it's not the one you think it is. But what this demonstrates is that given an identical stimulus, we see a wide range of responses in the human body. And it's this inter-individual variation that we see when people are exposed to low level of oxygens that really interests us from a scientific perspective. It's what we see at altitude, and it's what we see in the patients that we treat on the intensive care unit. So just to revise the sort of classic adaptations to hypoxia that we call acclimatization, there are three main processes that occur in three different body systems in humans. And some of you may have experienced these having gone to altitude yourself. So when you ascend to high altitude, your rate and depth of breathing increases to get more oxygen into the body. Your heart rate increases, which increases your cardiac output or the flow of blood around the body. And if you spend enough time at high altitude, the number of red blood cells increases, so your hemoglobin concentration increases, so you can carry more oxygen in the blood. Now, we have known this for many, many years. This is not exciting new information, and it goes some way to explain how it is that we can tolerate being at great altitudes for long periods of time. But it's by no means the whole story, because it is not the people with the highest oxygen levels or the highest hemoglobin levels that necessarily do best at altitude. So there must be more to the story than this. And just to help you with this oxygen delivery story, we can use this sort of classic analogy of a freight train just to try and think through the whole oxygen delivery message and what the answers might be. So here you can see a sort of freight train going around its track, and the lungs are represented by this loading station here. The load itself is the oxygen. Hemoglobin is represented by the trucks on the track. The sort of power of the heart, the cardiac output, is the train, and the tracks themselves are representing the blood vessels. So you can perhaps see that when there's a lack of oxygen, so not so much load available, by loading it quicker into the trucks, having more trucks and a more powerful locomotive, we can overcome some of the problems. And you can see this in data that we've collected, and these are data that we'll come back to in a little while in more detail. And basically what this graph shows here is some of our climbers ascending to the summit of Mount Everest, and we've taken arterial blood gases from them and measured the amount of oxygen and hemoglobin in the blood. So on the left, you can see the levels at sea level and getting progressively higher up the mountain to a maximum of 8,400 meters. And it's just the concept of oxygen delivery I want you to see here, and we'll come back to the numbers later. So what you can see is this progressive fall in the partial pressure of oxygen in the blood of these climbers as they ascend, a progressive rise in hemoglobin as part of acclimatization, and then this measure that we make, which we call oxygen content, which is the convective flow of oxygen around the body. And this is the bit which people also find a little bit hard to get your head around. There's actually a similar amount of oxygen flowing around the body at altitude than there is at sea level. It's just very difficult to get hold of it. And you might think it's a little bit pedantic, but I just want to dwell for a second on this. And for years and years, physiologists and clinicians have talked about oxygen delivery. Now, I don't know about you, but when I order something from Amazon, I'm not happy until that delivery arrives in my hand. And what we're talking about here with oxygen delivery is not really oxygen delivery at all, it's oxygen dispatch. So what I've just explained to you is the dispatch of oxygen from the left side of the heart, but goodness knows what happens to it after that. And it's useful terminology to think through in your mind, because quite a lot of what I'm going to explain during the rest of the talk looks at this sort of disparity between delivery and dispatch and the arrival of oxygen at the place where it needs to do its work in the organs and cells of your body. And dwelling on the freight train analogy again, we'll just pause from high altitude and just come back to the intensive care unit, because you might wonder, having seen this freight train analogy, and say, well, I know all about this intensive care stuff now, and this doesn't look very hard to me. If you've got a person who hasn't got very much oxygen in their heart or their lungs or their brain, then why don't you just increase all those things around the track and everything will be a lot better? Well, we thought that for many years, and we tried it, and we did a lot of studies, and in the end, we found that it didn't work. So giving more oxygen to people does not work and is very harmful to patients. Giving more blood to people does not work and is very harmful to patients. And giving them what we call inotropes to increase the cardiac output can work in some cases, but is harmful in other cases. So we have lost most of the manipulation on this oxygen delivery side of the track analogy, if you like. So we need to think a bit harder on the intensive care unit about how we can go about improving oxygen use in our patients. And just focusing on oxygen for a little bit longer, it sometimes comes as a surprise when I talk to people, particularly doctors, that oxygen might actually be bad for you. Due to the fact that we're breathing it currently and it doesn't seem to be doing us any harm, it's often been taught in years gone by that when patients are very unwell, if you can think of nothing else to do, just give them a lot of oxygen and it'll be okay. But if you look back very carefully to Joseph Priestley's work, and Joseph Priestley was one of the key figures in the discovery of oxygen in the late 1700s, and if you sort of decipher what he says here, I'll point out first of all that Priestley was a sort of odd chap, and he called oxygen dephlogisticated air, which was a bit of a mouthful and never really caught on. And what he said, though, was very important. After the experiments that he'd done, he stated that for as a candle burns out much faster in dephlogisticated than common air, so we might, as it may be said, live out too fast and the animal powers be too soon exhausted in this pure kind of air. So he basically discovered a pure kind of air, which was oxygen. He worked out that it was essential for life, but he said, there's something not quite right about this oxygen thing, and I think that if you breathe too much of it, it will be very bad for you. And about 100 years later, two scientists did some very similar experiments but looking at different outcomes and managed to show that oxygen was indeed very harmful. One of them was Paul Baer, who looked at neurotoxicity from oxygen, and the other was James Lorraine Smith, who in his laboratories in Belfast, Queen's College, did a series of experiments on mice using higher and higher concentrations of oxygen. And I'll just take you through a part of his experiments now. So he took two mice, put them in just over 70% oxygen or so for a grand total of 4 days, and by the end of that 4 days, one mouse was dead. And he thought, wow, this is not good. So he then did another experiment where he used 80% oxygen for another 4 days, and at the end of that, one mouse was dead. So he concluded from that there's at least a 50% mortality if you give mice 70 to 80% oxygen to breathe. But he also went on and did autopsies on this mice and very cleverly saw that what the oxygen was doing was causing an intense inflammatory reaction in the lungs. And we see the same inflammatory reaction in the lungs of patients and healthy volunteers when you give them very high concentrations of oxygen like this. And there's one more thing that I just want to introduce you to, another theme before we start moving on to some of the experiments at high altitude, and it's these little fellows, which I'm sure many of you recognize, and these are mitochondria. And these have become quite a focus of our research now and are fascinating. For those of you who've not met them before, these are the tiny little organelles. They're in almost every single cell in your body, and in fact almost every single cell on the face of the Earth. And their job is to convert energy. So they use oxygen to basically extract the energy from the food that you eat and turn that into the basic building blocks of energy for your cells, ATP or adenosine triphosphate. And they do this primarily through a process called oxidative phosphorylation. So an electron wiggles its way along 4 little complexes that we'll have a look at a little bit later, and in doing so, generates an energy within the mitochondria. And if you'll bear with me just for a moment, because I find this fascinating, so I'm going to force you to listen to it. The history of the mitochondria is really quite interesting indeed. The Earth is about 4.5 billion years old, and life started quite early on Earth, so about 4 billion years ago. There was no oxygen in the atmosphere 4 billion years ago, almost none whatsoever. But life continued in very simple-celled organisms until this point here, where life worked out how to harness the energy from the sun using photosynthesis, so using carbon dioxide from the air and light from the sun to generate energy with a toxic waste product called oxygen. And it was only at this point that oxygen started flooding the atmosphere. And within about 500 million years or so, the oxygen levels were rising rapidly on Earth. Some of the very primitive bacteria had worked out how to use oxygen to generate energy, but when the levels started going up and up and up, it started harming these bacteria. Something happened in nature which has rarely happened. I don't think there are any other examples of it. A process called endosymbiosis. So a larger bacteria engulfed one of these smaller oxygen-using bacteria, but rather than breaking it down, the smaller bacteria just remained inside the larger one. And when the larger bacteria replicated, the smaller one stayed within it forevermore and became your mitochondria. So the mitochondria within your cells today are sort of, their ancient ancestors were free-living archaea about 2.5 billion years ago. And if this hadn't have happened, there would have been no multicellular life on Earth at all. And just to focus just for a moment on this process of oxidative phosphorylation, because it's important for that story and for what comes ahead. Don't worry about the details of it. What is important in oxidative phosphorylation is these bits here. It's a normal part of this energy production process in the mitochondria to create these intermediate species known as oxygen-free radicals or reactive oxygen species. These are the superoxide anions. And what they are is basically an oxygen molecule with an extra electron on the outside, which makes it highly reactive. And what it wants to do then is just go around breaking things. In very low levels, the body can tolerate these. They have useful purposes in the body, and we can counteract them with innate antioxidants. But if the level of these reactive oxygen species gets too high, they have the ability to destroy almost every structure within inside a cell. They destroy lipids, protein, DNA, and they destroy the poor old mitochondria that create them. And the more oxygen that you provide to cells, the more reactive oxygen species they create. Okay, so we're going to go back now and continue our journey to high altitude. And you still may be wondering why, perhaps, why is this guy still, what's up with him? Why does he want to use high altitude as a model for critical illness? I still don't see the link. Perhaps the low oxygen thing, maybe there's a story in there. But actually, when you start digging deeper into both of these cohorts of individuals, you see a remarkable number of similarities between the two of them. This oxygen paradox that I have described, whereby they lack oxygen, but you give them more means to carry oxygen and it does nothing. We also see in both situations dramatic weight loss and wasting of muscle, both at altitude and in our critically ill patients, sleep disturbance, problems with cognition, and profound metabolic changes. So we think this is a reasonably good model for critical illness. Now, there are other models out there, and I'm conscious of the audience that I'm around. And the one thing we can say for sure is that animal models have repeatedly failed to deliver when looking at very complex pathophysiology like critical illness. Great for single defined diseases, but when you've got something as complicated as critical illness, certainly from the research that we look at, the animal models fail and fail again to bring forward any great advances for us in critical care. Also, looking at cultured cells, they're great for looking at single mechanisms to help bring the whole field forward, but not very useful in for us to understand a complex human response to a very complex disease process. And much of the literature, as I'm sure you've know historically, is quite muddled here now because of the ambient concentrations of oxygen used when culturing cells and describing hypoxia, normoxia, and hyperoxia. So whilst our model is not perfect, I think it stands up there amongst other models, all of them with their advantages and disadvantages. So over the years, we have conducted a number of studies at high altitude, starting in 2003 with a very simple trip to a mountain called Barunsi in the Himalayas, and progressing to larger and larger expeditions as time has gone by. The two largest of these expeditions were in 2007 and 2013, and they're the ones I'm going to concentrate on because I don't have time really to go through all of the expeditions. But so most of the data I'm going to show you has come from these two endeavors. So in 2007, what we set out to do was to try and construct the largest ever high-altitude experiment performed. And what, as I said before, we wanted to focus on was the inter-individual changes seen on ascent to altitude. So there have been many studies in the past taking small groups of people to high altitude, but never had there been a study of this magnitude to try and look at the differences between people as they ascend. In total, we took 222 volunteer members of the public to Everest Base Camp, which is at 5,300 meters above sea level. Of those, 14 were climbers and studied up to a maximum height of 8,400 meters. In addition, there were 60 investigators in the field running the laboratories and the experiments over the entire spring season in 2007. And in May of 2007, we managed to successfully put a team of 25 climbers onto the summit of Mount Everest, a combined team of Sherpas and our own scientists. One thing that we've maintained during this expedition and all the expeditions that followed is that we wanted to have an identical ascent profile for all of the participants. This is something that doesn't always happen in these types of experiments, because we wanted to be sure that everyone was exposed to the same environmental pressures by ascending identically. And what we had here is that everybody was tested prior to leaving for Nepal in our laboratories in London, and then tested 4 further times on their way up to Everest Base Camp, a trek lasting approximately 11 days. And for the climbers, there were 3 more laboratories set up on the mountain at Camp 2, Camp 4, and a very tiny place, only for an hour or so, called the Balcony, which we'll come to, where we continued to experiment on one another as we ascended and descended the mountain. During the course of the year that preceded the expedition, we spent an enormously long time preparing. We had a huge amount of equipment to take with us, and we wanted to make sure not only that it just worked, but it worked in a valid manner in the environment in which we were going to take it to. So you can see here some of the testing that we did in the year before going to Everest. On the top right there, you can see during the summer of 2006, we hired a freezer van and parked it outside our laboratories, and we're jumping in and out with sort of big down suits on all summer, much to the dismay of the people of Archway, testing the equipment and making sure that it didn't fail in very cold environments. And on the bottom left here, you can see a colleague of ours who's a hot air balloonist and was the first person to balloon over Everest. And he's a farmer in Suffolk, and he built this quite primitive hypobaric chamber on his farm, but it allowed us to do some testing of our equipment during that time. Not only did we test things in the laboratory and in these sort of environmental conditions, but we actually also ran 2 expeditions to this mountain here, which is Cho'oyu, the 6th highest mountain in the world. So in 2005 and 2006, we sent teams here to test the equipment, and not only that, to test ourselves to make sure that we were able to climb a mountain of this magnitude prior to going to Everest in 2007. So in the end, in 2007, we took 26 tons of equipment with us, all of which had to make its way up to the various laboratories, all the way up to 8,400 meters, an enormous task for our logistical team, with equipment moving by helicopter, these old-fashioned Russian helicopters, which have all now fallen out of the sky and been replaced, unfortunately, by person and by yak. It's always very difficult filling in a grant application and explaining how many yak herders you'll need for the next 3 months, but I think it caused much entertainment for the reviewers. And this is what the base camp of Mount Everest looks like in peak climbing season. There can be anything up to 1,000 people living there, so much busier than you might imagine. It's on the sort of glacial moraine of the Khumbu Glacier, sort of the white glacier you can see in the background. So actually, all of the tents that we put up are still actually moving on the ice beneath them. And you can just see in the top picture 3 of our big green military tents there, within which we have our laboratories. And you can see from this photograph that the standard of what we try to produce at altitude is pretty high. We try to replicate our laboratories at sea level, creating a physiology laboratory that anyone could just come into and work with huge amounts of equipment, heating, 24-hour electricity, etc. And during that expedition, we conducted over 40 simultaneous sets of experiments, and I'm just showing, I have no time at all to go through all of them, but just some pictures here to give you an idea of the kind of things that we were doing. On the top right, you can see some neurocognitive testing. On the bottom left, we're looking at the back of people's retinas to look at hemorrhages in the retina. And on the bottom right, we did a lot of exercise testing, so putting our volunteers on these cardiopulmonary exercise testing bikes and exercising them to maximum exhaustion all the way up to 8,000 meters in total. Further experiments here, all sorts of different things, amassing huge amounts of data. On the bottom right, a rather perilous experiment that we actually all decided we shouldn't do in the end. The expedition leader, Mike, on the bike here is looking very worried at me because we're just about to basically exsanguinate him at base camp after summiting Mount Everest to see what would happen if you suddenly dropped someone's hemoglobin level from about 20 to 15. And then we worked out they would actually probably be really bad. And we so we didn't do it in the end. So we were sensible about some things. A vast number of blood samples that we took during this trip. In this trip alone, we came back with 15,000 plasma samples, all of which were stored on liquid nitrogen. Over 100 gallons of liquid nitrogen were carried up to Everest Base Camp to facilitate this. Okay, so I'm going to share some of the results with you now, and I'm going to start with this experiment, which formed one of the chapters of my PhD. And the purpose of it were not particularly scientific, but really to draw everyone's attention to what we were doing. We wanted to try and quantify the limit of human tolerance to hypoxia. And the way in which we did that was to, in a small group of the climbers, to take arterial blood samples from one another and analyze them to measure the amount of oxygen in the blood as we ascended all the way to the summit of Everest, with the plan being to take a blood gas on the summit of Everest. Obviously, this had a few obstacles to overcome before we could complete the experiments, one of which was climbing Everest, and another of which was finding a system which would work at such high altitude and the ability to take bloods and to accurately measure what we wanted whilst up there. This is a sort of view of Everest that not so many people see. Those tents in the foreground are our tents. The bigger one in the middle is our laboratory tent, and inside there is a fully working exercise bike where we were doing VO2 max tests, so tests to maximum exertion at 8,000 meters. And what lies behind is called the triangular face. This camp is at 8,000 meters, delightfully known as the death zone. I don't think that needs too much explanation. And the triangular face behind it, you sort of pick your way up through the snow and rock, and the summit lies a bit further on from there. These are some of the members of our team, and actually 3 of the 4 people that the blood gases were taken from in the end. A very multidisciplinary team. On the left is Vijay, who's one of our medical students at the time. Then is Professor Chris Imre, a vascular surgeon, myself as an anaesthetist, and on the right, Sandeep Dillon, who is a general practitioner. And the 3 of us on the right are 3 of the 4 subjects from the blood gas. And you can see this is the triangular face again behind, with the clouds rolling in, as they often do in the afternoon up there. And you may not know that we summit these mountains basically in the dark, so that we have a long period of time in the day, the next day, to get down again, which is when the danger really occurs. So this is preparation at the South Col at about 9 p.m. Before summit day. And I show it because you can see all the equipment we're wearing, these big down suits and the climbing harnesses and ropes, and the only place that we could find that we could repeatably take blood gases from successfully was the femoral artery, which for those of you not good at anatomy is about here. So a complicated process of then having to strip off on the summit of Everest in order to expose our groin to one another for the femoral arterial stab. It's a truly phenomenal experience climbing Everest, and this photo was taken at about 4:30 in the morning, just as the sun was rising. You can see some of the bigger peaks of the Himalayas in the background there. And it's great because it brings sunshine and warmth with it, but it's also quite frightening because you realize how isolated you are and how cut off from every other human being on Earth. There is basically no rescue from this point upwards. This is the famous Hillary Step, so probably about 5:30-ish in the morning now. These 2 rocks here being the last real obstacle before reaching the summit of Everest. And this group of people here, just slightly before the summit, is our climbers, as we all agreed that we would summit together in one group. And we reached the summit of the mountain at about 6:30 in the morning on the 23rd of May, 2007. And whilst we were up there, obviously, we had this experiment to do. So we sort of did the sort of usual things that people do and then spent about 45 minutes trying to work out how to do the experiment. The problem was that it was very windy that day. It was marginal at best for climbing with about 20 to 25 mile an hour wind. And we thought that was probably not the best conditions to take our clothes off in. So we dropped back down the southeast ridge here, back the way we came, looking for a little flat sheltered area, and got to this area here where they called the balcony, erected a small survival shelter, and in pairs went in here and took the four blood gases, which are safely held here. This is Pasang, one of our Sherpas, in this flask, ready for him to take them back down to the blood gas machine, which is at 6,400 meters at Camp 2. We analysed those in a full benchtop analyzer, which had been specially adapted for working in these environments. And I'm just going to pause for just 1 second because I appreciate that not all of you may be used to looking at arterial blood gases or even oxygen saturation probes. And basically, if you put an oxygen saturation probe on a person, the highest number is 100, the lowest number is 0. So 100, anywhere between 95 and 100 is normal. And basically, from a hospital perspective, if you get down to the sort of 80 to 75 range, you're in a lot of trouble and you're probably going to end up coming to see us on the intensive care unit. And in those blood gases, so measuring the partial pressure of oxygen, that's equivalent of somewhere around 5 to 6 kilopascals. So these are the four blood gases that we took from one another, and just to summarize, the average partial pressure of oxygen was 3.3 kilopascals, which gives you an oxygen saturation of 54%. Subject 2 had particularly low values, as usual. They always perform like that. And their partial pressure of oxygen was 2.5 kilopascals with an oxygen saturation of 34%. Now, that was my blood gas. Which may or may not have caused some damage to me, I don't really know. But with those very low levels of oxygen, I could still operate normally, take arterial blood gases from other people successfully and first time. And it goes to demonstrate that humans can tolerate exceedingly low levels of oxygen if given enough time to acclimatize. But those are all very well and a great headline news, but it's perhaps not entirely useful to know how much oxygen there is on a climber stood on the top of Everest, because it's a pretty rare event. But remember, we have now taken hundreds of people actually to Everest Base Camp to study them, ordinary people like yourselves who've just volunteered to come along. And these ordinary people here, as an example, are my parents, and they came along and were experimented on just like everybody else. And when you look at the oxygen saturation in people at Everest Base Camp, it's around about 80% on average. And if you do a blood gas on them, the partial pressure of oxygen is about 6. And it's Base Camp that we're doing most of our work at and most of our experiments. And it's this kind of comparable to the level of hypoxia that we see in patients who are unwell in the hospital, and particularly the intensive care unit. So I'm going to return to this analogy again and move us on a little bit, right to the very end of what we call the oxygen cascade now, to something that's called the microcirculation. So oxygen delivery, as I said, sort of explains the convective dispatch of oxygen from the heart, but we have no idea what happens after that. And we've become very interested in what's called the microcirculation, which are the very smallest blood vessels in the body, the arterioles, the capillaries, and the venules. And it's in the capillaries where oxygen diffuses out into the cells to do its great work. And we've been using a system, a microscope system, that allows you to visualize the microcirculation in humans. We tend to look at the sublingual microcirculation under your tongue, not particularly because it's of interest to us, but primarily just because it's easy to access with this camera. We are able to generate images like this from this camera, and you can see here in these capillaries individual red blood cells traveling through capillaries, and we can then load this into some software, and from that calculate density of vessels and the speed of the blood flowing around the capillaries. We see very interesting changes as we ascend to high altitude. On the first hand, the density of vessels increases, which you might expect. But what was a surprise to us is the speed of the blood going round slows down. Now, we had predicted that it would speed up. If you are lacking in oxygen, you might find that the blood flow is faster to deliver more oxygen in the tissues, but actually it slows down quite markedly. And the longer you're at altitude and the higher you go, the more it slows down, which was a bit of a surprise to us. And back to this analogy again, moving one stage further, we felt we should delve right into the end of the oxygen cascade and look at where oxygen is used by the cells and how that is changed at altitude. We first started doing this using a technique called magnetic resonance spectroscopy, which uses an MRI scanner like you might find in a hospital, which has got some special software in it so we can look at molecules as well as images. Now, whilst we may have achieved some pretty amazing things at altitude, we've yet to be able to carry an MRI scanner all the way to base camp. So what we had to do was scan people immediately before they went to altitude and literally the sort of day they came back in their return to the UK. And we were particularly interested in this experiment looking at the heart. So we scanned cardiac cells and we used a measure called phosphocreatinine ATP ratio, so looking at the energetics in the heart. And you can see in that middle bar there a significant reduction in the amount of energy available in the heart at high altitude, which thankfully, as I was one of the subjects, recovers again when you 6 months later after you've been at sea level. And at the same time, you see a reduction in the function of the heart, particularly in the diastolic, so the relaxation phase of the heart's function. But really to look at the cells that we want to look at, there's no other way around it but we have to actually take cells from people. And the kind of thing we want to do, you can't really look at in blood samples. So it involved us taking muscle biopsies from one another. And you can see here the procedure that involves. I have a nice collection of scars I could show you afterwards if you ask me nicely. And we do a whole range of things with these muscle samples. One of the things we do is called proteomics, so we can look at the type of protein in the muscle and how much of it there is. And I'm not for a minute going to dwell on any of this complex biochemistry, but the important thing to say is all across the whole energy production process in the muscle, everything is downregulated. So through glycolysis, through Krebs cycle, and oxidative phosphorylation. And then we move on to the mitochondria, the final users of oxygen in the body. And this electron micrograph here has been stained so that you can see the mitochondria. They're the dark spots there. And this is the muscle at sea level, and this is the muscle after about 2 months at high altitude. And what you see is dramatic reduction in the number of mitochondria in the muscle, again, opposite to what you might expect if you were lacking in oxygen. We can also look at the function of those mitochondria, looking at those complexes in the membrane that I showed you. And again, we see downregulation in those complexes, along with a downregulation of something that's called uncoupling protein, which suggests an increase in the efficiency of how the mitochondria are working. And one of these translational coactivators. So basically, the whole of the gene mechanism of this skeletal muscle being down-regulated. We're also very interested in a small molecule called nitric oxide, not to be confused with the molecule that we use as anaesthetists, which is nitrous oxide or laughing gas, a very different molecule indeed. And nitric oxide is a regulator of many, many pathways in the body. And we were particularly interested in it because it's related to blood flow, because it controls smooth muscle. It has an effect on the mitochondria and on reactive oxygen species. It's very difficult to measure the amount of nitric oxide in the blood, but we can look at its fingerprint through nitrate, nitrite, and something called cyclic GMP. And sure enough, we see an upregulation of all of these in our climbers as they ascend to high altitude, meaning that perhaps this nitric oxide molecule is very important in the acclimatization process and perhaps controlling the mitochondria and controlling the microcirculation. And as my journey begins to come to an end now, I just want to introduce one more theme. And you will be very familiar with this iconic photo that was taken in May of 1953, the British expedition which put a New Zealander and a Nepali on the summit of Mount Everest. And what a lot of people don't realise that this is not Sir Edmund Hillary, this is Tenzing Norgay. It's rumoured that Tenzing could not use the camera, so there is actually no picture of Hillary on the summit. So when these pictures were beamed back to the British public, for many of them, it was the first time they had seen or heard of Sherpas. And any of you who've been to the Himalayas will have met these people and been in awe of what they can do at altitude. They move around and climb at altitude as if they were at sea level. Their performance is astonishing. Now, they are descended from Tibetans who have been on the high Tibetan plateau for about 20,000 years now. So they've clearly inherited something that allows them to perform well at altitude. But what that translates to from a physiological perspective remains unclear. So we really don't understand much about the Sherpa physiology at all. Going back to our freight train analogy, if you were trying to work out why the Sherpas might be fitter than us at altitude, you might suspect them to have a better cardiorespiratory system, to breathe more, to have higher oxygen saturations. That is not the case. Their oxygen saturation is always the same as ours. You might expect them to have a high hemoglobin level, so be able to carry more oxygen. That is not the case. Their hemoglobin is always lower than ours at altitude. And you might expect them to be physically fitter than us, and they very definitely are not. So this prompted us to go back to Everest in 2013 and put on another large-scale expedition, about 180 participants in total this time, but really focusing now on studying the Sherpas. And 65 of those 180 participants were Sherpas who came to Everest Base Camp with us. We kind of went a little bit earlier in the season for various reasons in 2013, so Base Camp had a good covering of snow on it when we arrived. We conducted many experiments similar to the 2007 expedition with some additional ones, with the Sherpas following us step by step on that identical ascent profile, so directly comparing us to them as we ascended. We remained very interested in the microcirculation, and you can see the microcirculation of some Sherpas here being measured with that camera device. And I'm not going to dwell at all really on the results, but just to say what you can see, this is the flow in the blood vessels that I showed you before, that the Sherpas are the blue and us are the open boxes. And just like before, you can see our flow slows down. But the very interesting thing is that not only does Sherpa blood flow not slow down, but it gets even faster at altitude. So much more blood flowing through all the tissues of Sherpas at high altitude. The blood vessel density, as before, our vessel density in the white here increases, but the Sherpas much, much more than us. So they have more vessels and more blood flows through them. We were also very keen to look at their mitochondria, so more muscle biopsies, and I had the sort of duty of stepping up to try and be a surgeon for the expedition, taking all the muscle biopsies from everyone here. The difference in this expedition was that previously we froze all the samples, took them home and analysed them in the laboratories. This time round we took a lot of the equipment to Everest Base Camp with us, which had never been done before, and used these devices which are called respirometers to look at live muscle tissue up at Everest Base Camp to understand what was happening to the mitochondria. In brief, we take the biopsy, we separate out muscle fibers, we sort of punch holes chemically in the fibers, and then put them in this respirometer device, and then measure how much oxygen is being consumed. We add to that chamber a whole host of inhibitors and substrates and uncouplers to basically work our way through this process of oxidative phosphorylation to get enormous amounts of information about the live tissue that we're looking at. And I don't have time to go through in detail the results that we have, but the conclusions of them, and very exciting for us, were that the Sherpas very clearly have a much more efficient mitochondrial system than we do. So they can produce more energy energy for the same amount of oxygen. Part of that is explained by a substrate switch, so they stop using or reduce their use of fatty acids, which consume a lot of oxygen when you burn them to make energy. And they also seem to be much better protected from this reactive oxygen species oxidative stress than we do. Okay, so we're at the final stages of our journey now, and I'm going to bring you back down to earth with a bump and back to the intensive care unit and sea level. What I want to do in the last few minutes is just share with you a couple of studies that we're doing in our patients currently based on the work that you've seen here at high altitude. So the first of these studies, we wanted to emulate exactly what we did with the Sherpas and ourselves at high altitude. And those of you who embark in clinical studies will know that it's absolutely mandatory to have an acronym for every study you do. It's even on the form now. What is your acronym? You can't move forward unless you have one. So we pride ourselves in coming up with great acronyms for all our studies. It took a very long time to come up with this one. But what we did was to use that camera device again in critically ill patients on the intensive care unit to look at their microcirculation, to take muscle biopsies from them to look at mitochondrial function, take blood from them to look at nitric oxide activity, and then follow the course of these critically ill patients through the intensive care unit, taking further samples as we go. Now, this is our laboratory here, and Dr. Helen McKenna, who sat in the front row, worked enormously hard against great adversity to get some enormous amounts of fantastic data from these patients. And now, paradoxically, I think that she had a harsher environment to work in than we did at Everest Base Camp. Because see this little desk here, UCL very generously gave us 3 foot of desk to work in. They didn't tell us until we got to this little area that it was quite high in the air. So poor Helen had to run this entire experiment for 2 years, stood on a stool. But we have some truly fascinating data. We hope to publish this soon, so I'm not going to go through it today. But some wonderful mitochondrial biomarkers of recovery in the critically ill, which ties in exactly with the data that we saw in the Sherpas. So the Sherpa and lowlander model at altitude has led us directly to this finding, which we're very sort of proud to have now. Another study we're running currently at the Royal Free Hospital and Southampton General Hospital, this whole premise that oxygen is harmful, we're randomizing intensive care patients into a normal level of oxygen or quite a low level of oxygen. Because not only do we think that it will do them no harm, but we actually think that having a lower level of oxygen is potentially beneficial to these patients. And we're measuring some of these markers of oxidative stress in those patients that we're randomizing to one arm or the other. And the other study that we're doing, we're sort of branching out now from intensive care and looking at patients who undergo major surgery. Many of these patients have complications and side effects which ultimately lead to them becoming critically ill and coming to the intensive care unit. And we're looking at the function of their mitochondria before and after surgery and looking at these oxidative stress markers. And Dr. Jar Stevens, who's also here, has nearly completed this study now, looking at a large group of patients having surgery at the Royal Free Hospital. So 3 clinical translational studies that have resulted directly from our work at altitude. So to round up, what have we learnt from high altitude? Well, I think hopefully you'll agree with me that this oxygen delivery story that we're taught at medical school and university is very important, but it's not the entire answer, because both at altitude and in the patients that we look after in intensive care, even if you normalize this all back perfectly, things do not go quite as you had hoped. And perhaps the answers in both situations, both at altitude and for the intensive care patients, we need to look much, much deeper into their physiology, into the microcirculation and beyond, into the cells, if we are to actually come up with meaningful treatments that can save lives. And where do I see the future of high altitude research? I'm very conscious there are a lot of young and enthusiastic scientists out there, and hopefully somebody can take this over from us one day so I don't have to go back to altitude again. And their technology is just truly fascinating these days. So much has changed in the 15 years that we've been doing this, lugging enormous great boxes up to altitude to get 1 or 2 readings. There is incredibly sophisticated technology now that produces very high-frequency data, all of the omics platforms that are available in the blood that we bring back from altitude. So I hope the generation that follows on afterwards can utilize these to really look much, much deeper into the phenotype of people going to high altitude and bring that information back to sea level with them. So with that, I'd like to thank all of our sponsors over the many years that we've been doing this, which include the Physiological Society. But in particular, I'd like to thank all of the trekkers who have come on these expeditions with us, freely giving of their time, their blood, and their muscle to help us to try and save lives on the intensive care unit. And thank you all for coming this evening.